Ringens talks, so I'll see what goes on. And there's a reason I'm saying that. But there's a whole bunch of us who are interested in Mimulus. And what you're going to discover is that if we had been able to coordinate with the talk organizers, my talk would have preceded Wendy's really nice explanation of what we will do once you find something out. And I'm now going to tell you what we found out. Sorry that it didn't work that way, but we couldn't manage to swap those around. Selfing is a really important part of biology for a whole lot of plants. I don't think I have to tell most people in this room a whole lot about that. But it's something that we often talk about as being a species characteristic. And so this species is a selfer, this one's an outcrosser. And that's not a bad way to start off. But if you look a little closer, as Wendy nicely pointed out, within any one species, some of them go from they always cross to other populations that always self. And there's something going on there. And understanding the causes of that is something that would help us to recognize how much of this is just ecological noise, as my theoreticians friends would like to say, and how much of it is something that's due to heritable traits, as Wendy so nicely explained. It's useful to break this down a little bit and think about in a little different way than Wendy did. It's easy to imagine, if there's no pollinators, the plant has no choice, and all the pollen that arrives on the flowers are going to wind up being self, and then there's not a whole lot of choice. The plant, if it is self-compatible, is going to be only reproduced in that way. Likewise, if there's a whole bunch of pollinators, there's a lot more opportunity for crossing. So these ecological factors, and there's a whole bunch of other ones that might be important, could really play an important role in generating what we can measure in the field when we do the hard genetic work to figure out what's going on. But genetic factors can also play a role because of this weird feedback that happens, because those seeds that come out might show inbreeding depression, and therefore not be as valuable to the fitness of the plant. And that's going to feed back here. So what we're really interested in are there traits of the plant that are inherited from one generation to the next that are affecting that. That's what's going to allow us to generate, there might be variation among plants that's due to these heritable attributes, and that would mean that we could have, this one is evolving to be a selfer, and this one evolving to crosser, and now we've got the startings of real evolutionary change. So we've done work on that. There is all kinds of work that's been done on Mimulus ringens. This is just yet another little corner of what I want to point to. We can look at both genetic, some factors that are likely to be genetic and some factors that are more likely to be ecological. And what's really interesting is that both of them types seem to play a role in this, and I'm sure other species, but they interact with one another. For example, here's display, which is a trait that may well be under genetic control, and how many flowers a bee probes before it departs and goes to another plant, allowing the opportunity for outcrossing to occur. And if a plant's got more flowers, the bee spends a lot longer time, and it's going to be causing more and more selfing to occur. And son of a gun, that's exactly what happens when you actually go and confirm that this ecological field behavior that might be caused by a heritable trait is going on. So there's sort of, it's a very indirect connection that's going on. But this is, to get this is, most of you may have tried this. This is hard. The only way you can get at this kind of data a lot of times is to do it in those wonderful square grids we've been setting up that you've seen most of the day here. There's a reason we did them, because we can get data. Doing it in the field is much more challenging. But it is also very, very valuable, because it could be that those ecological factors dominate this, and we're all sort of building sandcastles to try to explain what's going on. So we need to start to get it into that diagram and start to isolate it. So this suite of approaches where we're trying to look at things in a very tightly controlled, carefully precise way, but then something we haven't done a whole lot of up to now is to back off and say, okay, so what's going on in some real populations? And recently we got enough money from NSF to do that, because that's really the limiting factor. And so I just want to, I could do. Give me some rubber boots and a piece of paper, and I can do most of this work, because the bees are very cooperative. But I can't tell who the father of the seed is, and until I can tell that, I won't know the selfing rate, but it also all that male function stuff, because that's where half the genes are coming from. Everybody's got a mother and a dad, even a seed. So we got that, and that is a limiting factor for all this. And so what we just wanted to do some field surveys to try to get an idea of what's going on. So these are meaningless names, but here's real meaning names, Akron and Cleveland, very important places that you need to know about. And why do you want to know about Cleveland?
Because of the meeting next year.
The meeting next year. So you're going to come visit me, okay? And several other people. So we want to see, now you already know the answers to this, right? Because this is going to be call and response here. Just so you know most of what's we want to know whether there's variation, and you already know the answer, and then some of the factors associated, and you don't know that yet. So here we are. We're over here somewhere. Now we're here. This is to give you an idea of there. I get to go out to these beautiful wetlands, see these really cool plants. We chose 13 sites because that's all I could handle. I wanted to do more, but couldn't. And we chose them very specifically because we think floral display is a key trait here. So I actually went out the year before and I found places where the plants had a lot of fruits on them, and I found places where the plants were smaller. So I was trying to maximize that dimension. And then I measured everything I could. I'll tell you about a few of them. The rest you'll have to hear about in Cleveland. So here's a sideways version of the figure that Wendy so nicely explained to us. These are things she has already told us. There's a whole bunch of variation with the 20 to 65% variation she's talked about. And you can see that there's a whole lot of there's no indication that the eastern half is all sulfurs, which might happen if there was an invasion of a selfie variant or something like that. So we now sort of covered a big part of Wendy's background that sets her up for a really cool study that I'm really excited to be helped with. You can also, thanks to Dorothy Christopher's expertise with this, we can look at the genetic relationships among these populations to see whether there's any indication that maybe, although they're not spatially segregated, there's just a sort of a lineage of selfers that's come in, or a lineage of outcrossers that's come on. Here's our most outcrossing, and here's our most selfing. They're among the most closely related types. So that's not the explanation either. This is a bunch of independent variation. Now, that could be independent ecological variation, or it might not. So let's see what's going on with that. Let's look at display. And you can see that it varies a whole lot. I've tried to use colors to match these sites because these letters are meaningless to anyone but me. Although, this is a really sweet site. Each site, I went out there every 2 or 3 times a week to 81 marked plants and just counted how many flowers were being shown each day. And then for each plant, I took the maximum number of flowers it had on any one day as my indication of what it was showing to bees. And it varies a whole bunch. It is not really spatially clustered in any meaningful way. You can see the dark colors. There's a couple of them here, but there's darker ones over here. We can also look to see whether that maximum floral display is associated with the selfing rate. So now this is supposed to have been the stuff that set you up for where Wendy was going. And it's too small. I'm not going to do statistics for you today because we still got half the data to come. And I don't want to have to go back and say I was completely wrong. So I'll tell you what we see so far is a hint that maybe something's going on, that plants in these field populations are responding the same way that plants in our controlled environments are responding that way. And so there's some indication that an important attribute of the plants that's probably got at least some genetic contribution to it is in play for affecting selfing rate. We can also look at, we did this independently, but arrows seem to be the way you show corolla with. There's a lot of variation. It's sort of, but look, some of them, they're down here 17, these are up there 25. A lot of variation, but a whole lot of sites that are almost identical in their flower size, which I think is really fun. In a separate study, we did broad sense heritability, measures just clonal repeatability, and this is a highly significant amount of heritability. Something's real going on there. And so we know that this trait is something that's, although there's likely to be some environmental effect, it's one of those genetic factors. And it's not really spatially segregated in any meaningful way. If you have a good idea other than autocorrelation analysis, figuring out whether there is a spatial pattern, talk to me. And we get, yet again, a sort of a interesting preliminary idea that maybe the size of the flowers might be affecting the selfing rate. And so where that you can't see these horizontal standard error bars some of the time because that's the variation. Except a few cases, this one site we have only, I think, 13 out of 20 plants in so far. So the vertical, the selfing rate, we got tight estimates on selfing rates. So we're pretty confident that it is what happened that particular time. And so when you don't see them, it's because they're hidden. So we got a pretty good indication that big flowers tend to outcross more, which is very satisfying. I can do the same graph for corolla height, for anther stigma separation, for a number of other things. Some of them are most of those are a little weaker than this one. I'm going to show you the best data I got so far, but all of them show a hint that there's probably structural and functional connections between the morphology of flowers and the amount of selfing that's going on. But doing statistics with only 6 data points and 6 predictor variables, it raises some complications that I haven't yet figured out yet. So we need more data. We also looked at pollinators. So I went out there and I measured who was visiting. And what I've done is I've ranked the sites in yet another order. But what you need to look at is that some of them have no yellow or orange coloration, indicating that they have no bumblebees and they have no medium-sized bees. That's honeybees, very rarely, Anthophora abrupta, and a couple of others that show very rarely. It's bumblebees. And so there's wide variation in the abundance of these bees. And so I plotted that down here, and I didn't even bother putting the standard errors in right now because I was short on time to get ready for this. But they're bigger than is indicated here. And the selfing rate, and there's not really anything going on so far. I can make it work. I have to do a lot of extra things. So that's not going on. But I also noticed something we have not seen much in our controlled environments, which is that although there we almost only see bumblebees, we're seeing these little green dudes, gals. Those little serotinas, lazy glossum, some things like that. And they're getting in there, and they're being what James Thompson has so wonderfully called ugly pollination. There's good pollinators, like bumblebees. There's bad pollinators that sort of get in there, but they don't move much pollen. There's ugly pollinators that not only don't move pollen, but they steal it. They're pollen thieves. And that's what it looks like they're doing. And there's something very interesting going on with them, and that's what I'm doing this summer. We don't have money to do any more selfie estimates, so it's going to be tricky, but I'll see what we can do. So we think something's going on with this part of the ecological world. So the conclusions are the wrong word, but what we've got so far, the preliminary results, that's what I said. There's certainly a lot of variation going on. It looks like there's some things that are genetic and some things that are more ecological that are affecting the variation out there. And now the thing to do is to get in there and figure out not just are some of those working, how much of each? How can we do this to understand what's going on in real wild populations of this cool plant? Thanks. Yes?
I'll be a bit provocative.
Oh, I
just
knew
for
that.
Fun. You know, I've watched this research program over the last 20 years, starting off with Jeff, and it's kind of interesting because you guys started off with, arrays, and now you're going to natural populations, which I think is great, but you're focusing on the among population variation. And I'm sort of wondering, having listened to Mike Whitlock's talk this morning about doubt, and I have, having estimated a fair number of selfing rates myself, about the accuracy of the selfing rates, and I'm wondering if you should sort of really seriously consider, one, using Boris, not MLTR.
Okay.
And secondly, going to individual measurements of individual plant selfing rates. You're just using a population estimate. And Britain will be the first to admit that those standard errors look tiny for a good reason. They're not. They're not actually really giving you a measure of.
I'm going to punt to Jeff because this is his bread and butter.
So we,
did you try
in our
Boris?
experimental populations,
Yeah.
so actually we've done it through paternity analysis,
Yeah.
right? So almost all of our previous work was paternity analysis,
Yeah, but this-
not MLTR.
Yeah, these
But
are-
I will tell you that we, like in our experimental populations, we get the exact same number with MLTR as we get with paternity analysis.
Yeah, but that's because you haven't actually a prescribed number of individuals that you're working with, whereas in a natural population, you're sampling 20 from a population that might have 60 individuals in it.
But
And so the variance in, you know, what's the variance in that population among individuals? I mean, there's error associated with.
But they are random. I did, honest to God, random sampling. I put out squares. I chose random things. It's.
Oh, I trust you.
No, but if that's the case, then we have a random sample that,
Yeah, sure.
that it should have the right characteristics.
No, it's always bothered me that
Oh,
you know,
yes.
when you do open pollinated families, you're taking a sample from a population, and that sample can be quite a small sample. And the question is, what's the error associated with that? I mean, you'd really like to know what is the variance among individuals in their self.
I agree completely. Yes.
It's just a thought, something you guys might want to do. You know, you go from arrays to among, and now you finish off with detail. Think of the animal people are all into individual-based measures. And
Have
we
you
don't
ever
do that
considered
in
reviewing
human
for National
systems.
Science Foundation when a grant like that?
Yeah, just. I mean, that's what John Kelly has been suggesting that we do, and that's why he developed forests, so that you could get.
Okay.
I mean, Kermit tried to get his method to give you individual estimates, but you need very large sample sizes per month. But with John's method, which is a Bayesian method, you actually don't need such big sample sizes. And I really think it'd be worthwhile trying to do, you know, maybe 1 or 2 populations where you mark all the individuals and you map them, and you get the T estimate for every individual. I think you'd really learn about the role of ecology by doing that kind of almost demographic analysis of the meaning system. That's good. Something to think about. Sorry. I like this stuff.
I like being pushed. I love it. The gong has gone, but I'm happy to answer more questions, or you can go and eat. It's fine with me.